Hello, respected fellows. I hope you are all doing well. I am Maham Riaz and from the Department of Zoology, Wildlife and Fisheries, University of Agriculture, Faisalabad. First of all, I would like to thank to the Applied Zoological Society of Pakistan to give me the opportunity to present my research, the evaluation of the toxic effects of the benzophenone on the histopathology of the Labeo rohita. As the title shows, evaluating the effects of the benzophenone. So the first question is, what is the benzophenone? The benzophenones are the organic ultraviolet filters that are widely used in the personal care products such as the cosmetic sunscreens and the aftershaves. Now the second question is how these contaminants enter into the environment to affect the aquatic organisms, particularly the fish. They are the two sources. First one is direct sources that include the industrial effluents, surface runoff, and the swimming. And the second one is the indirect sources that include the wastewater treatment facilities. Now, the how these contaminants affects the fish. Because the fish species are the excellent model for monitoring the presence of the contaminants in the aquatic environment. Because these species are susceptible to even the minor concentration of these kinds of the contaminants in the environment. So how we can evaluate the stress of the fish. So for this purpose, we use the histopathological indicators. These indicators are widely used in the fish health research because they provide the accurate information on the chronic and the sublethal effects of the contaminants on the fish organs. There are the several histopathological indicators that are widely used in the fish health research that are the kidney, liver, skin, and the gills. Due to the extensive use of the benzophenone in the personal care products, its concentration range in the environment from the nanograms to the microgram per liter. Here are the few stats. Stats that shows that its concentrations into the different aquatic environments. As we see that the maximum levels are reported in the liver, which are the 44 microgram per liter, followed by the seawater, wastewater, influent swimming pool, tap water, lakes, and the groundwater. So the minimum concentration has been reported in the groundwater, while the maximum concentration has been reported in the river. So the extensive use of the benzophenone has increased its concentration in the water, which raised the concern about its potential toxicity and its effects on the health of the aquatic environment and ultimately on the mankind. Now the question is why I selected the Labeo rohita as the test organism. As we know, the Pakistan is located in the South Asia, and this species is widely cultured in the South Asia, so it is easily available. And the second is this species, freshwater species, is important in the nutrient cycle and the trophic nutrition. By doing this, it plays an important role in the ecological balance of the aquatic environment. Now the next is what is the hypothesis of the my research. By considering the effects of the benzophenone on the other aquatic organisms and its toxicity for the fish, and considering that the fish are susceptible to the minor concentration of the contaminants, I make the hypothesis for my research that the benzophenone exposure cause the histopathological damage to the muscles and the gills of the labial heat. And now the question is why I use muscle and the gills as the organs, because these two organs are in the directly contact to the water as compared to the other organs. Before going to the methodology section, first of all, I would like to review the existing literature available on the toxic effects of the benzophenone on the aquatic organism, particularly fish. This literature proved that the benzophenone exposure is detrimental to the aquatic organism, and it showed that the BP exposure causes oxidative stress, neurotoxicity, apoptosis, cytotoxicity, toxicity, genotoxicity, endocrine disruption, developmental toxicity, and the DNA damage to the fish. It also exerts the negative impact on the social behavior of the fish, metabolic system, especially the lipid metabolism of the fish, and the cause the alteration in the blood cells. The histopathological analysis of the various organs, such as the kidney, liver, skin, gills, and the reproductive organs, show that it causes the negative impact on these organs. To perform my research, first I sourced the crystalline benzophenone and crushed it into the powder and dissolved in 0.1% dimethyl oxide, which is commonly known as DMSO solvent. Then I made the 100 mg per liter stock solution and stored it at the 20 degrees Celsius. Now the further dilution of the stock solution were the made to prepare the working solution according to the requirement. The Labeo rohita fingerlings were the source from the local hatchery and they acclimatized for the 10 days in the cemented tanks containing the reconstituted tap water. I performed my research in the two phases. One is the acute phase and the second one is the chronic phase. So for the both phases, I made the four experimental groups: two for aquatic, two for the acute exposure and two for the chronic exposure. And in the each group, the three replicators were the used. Now in each replicate, the 10 fishes were the used and each group contained the 30 fishes. So the total four groups contain the 120 fingerlings of the Labeo rohita. The control group contained the only the reconstituted water without the benzophenone. For the acute exposure, the concentration of the benzophenone this started from the 100 microgram per liter and gradually increased to the 1000 microgram per liter. And the time limit was the 96 hour. While for the chronic exposure, the time frame was the 35 days and the concentration was the sublethal concentration of the LC50, which is the 122 microgram per liter. The physicochemical parameters of the water were maintained during the acute toxicity through the different analytical methods, as you can see here, and the dead fishes were removed after the 24 hours to prevent the contamination in the aquatic in the test environment. This picture clearly shows that the whole experimental design of this research in the acute exposure, the 100 to the 1000 microgram per liter of the concentration were the given to determine the 96 or the LC50 and the LC99. And in the chronic phase, the fish were exposed to the sublethal dose for the 35 days, and after that, the fish were the euthanized and dissected to isolate their gills and the muscles, and these organs were histologically examined. These tissues were preserved in the formalin embedded in the wax and stained with the hematoxylin and the erosin, and its morphological and these tissues were examined and its morphological changes in the treated groups and the control groups were compared. In the acute toxicity of the benzophenone, the 96 or LC50 and the LC99 were determined by the probit analysis method using the Minitab software. These lethal concentrations were measured along with the 95% the confidence interval. This table clearly shows it LC50 95% confidence interval and the lethal concentration at its 95% confidence interval, chi-squared F value and the p value. The graph shows its increasing mortality of the Labeo rohita during the acute exposure. These are the histopathological slides of the treated and the normal gills. The A is the slide of the normal gill, which clearly show the normal histopathology of the mucus cells and the pillar cells. While in the B we can see that the black arrow showing the intralaminar debris, and the other straw show the cellular damage in the mucus and the pillar cell, which is confirming the toxic effects of the BP on the gills histology. Similar trends have been shown in the slides of the muscle histopathology, where the vascular necrosis, intralaminar debris, and the muscle fragmentation of the intact structures have been found after the exposure to the benzophenone. So, my research confirmed the hypothesis that the benzophenone exposure caused the toxic effects on the histopathology of the labeo rohita, and these results are the consistent with the previous studies that demonstrated the toxic effects of the benzophenone on the fish and other aquatic organisms. The finding of the my research also emphasized that by considering the increasing concentration in the aquatic environment, so there must be some regulation or regulatory control to ensure the safety of the water resources. But it has been noted that the international organizations, including the Environmental Protection Agency, have not established a specific permissible limit for the BPWV filter in the drinking water, which shows a huge vacuum in the regulatory framework for the specific chemicals. Thank you for your attention and listening me.